Thank you so much, everyone, for being here. I really appreciate your time and being in my audience. So firstly, I'm so grateful to be here on traditional, ancestral, and unceded territory of Musqueam, Squamish, and Tsleil-Waututh Nations. My name is Samantha Rhodes, and I am a graduate student at UBC, where I study in the Pacific Salmon Ecology and Conservation Lab. My research is on juvenile sockeye migration in the lower Fraser River, and I'm excited to talk to you about life history strategies of salmon and also drivers of change of their migration. So firstly, thank you so much to the supporters and funders of my research. It takes a lot of hands to do this kind of science, so I'm really grateful for their assistance. So I've done my undergrad at UBC in forestry, and I'm now doing my master's. And my favorite thing from studying the environment and also growing up on the North Shore is how nature really has a tendency to behave like magic. When we're little, we're taught that plants breathe out the same air that we breathe in and vice versa, and how the water that we drink has traveled all over the world in the water cycles. So studying salmon, I've really seen that they possess the same kind of magic, and I think that the migration is a really big example of this. So I know that we're all probably familiar with the life cycle of Pacific salmon, but generally speaking, there's some deviation between species, but Pacific salmon are anadromous fish. So this means that they have a freshwater and saltwater life stage that they migrate between for their reproduction. So generally speaking, salmon are born in the freshwater. The eggs are laid in gravel beds called redds. And then they grow up as alevins. They hatch out of their eggs with the yolk sacs still attached to their stomachs. And then they grow up in freshwater for a period of time that varies between species. For sockeye salmon, which is my focal species, they tend to stay in freshwater for about a year. And then they migrate to a new environment called the estuary. The estuary is the transition zone between freshwater and saltwater, and it's a really important area then for fish to develop in a way that they can then withstand the new conditions of the ocean. Once they're in the ocean, they have their adult life phase, and then they return to their natal streams or their home streams to spawn, and the whole cycle continues on. So while there's differences between species, there's also differences within species, and in salmon, that can be expressed as the life history strategies that they have. So a life history strategy is variation in a species trait or their life cycle that leads to differences between each other, but it has the ultimate goal of sustaining the fish's survivorship and also reproduction in the future. So what causes a fish to migrate in the first place? There are a few different drivers of this. So the first is a migration strategy that is a fixed migration strategy. These are called obligate migrations, and this kind of migration is caused by either a gene that the individual possesses or by a trait that used to be flexible, but with time and repeated choice in the species itself, is now become a trait that is expressed over and over again. So another kind of migration strategy is one that has to do with thresholds or conditions being met. So this one is called a facultative migration strategy. And we can think of this as being cued by milestones in development or conditions. So in ecology, we also talk about stable states often, and there being a tipping point. So we can apply that kind of thinking to this kind of migration strategy, where there's a tipping point in the condition that then allows the fish to migrate itself. So this sounds more familiar to what we would think of for salmon. Importantly, though, the environment is a really huge driver of migration in salmon as well. And that's because the environment the fish is in, like temperature, can impact the fish's development and their condition as they're moving through the environment. So in sockeye salmon, there are two different strategies that are within the species as their life history strategies, and this pertains to the age that the fish migrates. So the first strategy is called a yearly migrant, And the second strategy is called a sub-yearling migrant. So yearling migrants are believed to be the dominant life history strategy of sockeye salmon here. They follow the typical life cycle of salmon where they retain or maintain their existence in a freshwater environment before they move to the ocean. So these fish rear, meaning that they feed and grow in a freshwater habitat for about a year before they go out to the ocean. This means that the fish have had opportunity to feed and then also avoid predation that they could have faced in a larger habitat like the ocean. And so these fish are larger bodied when they start to move through the estuary. They also are expected to have an earlier migration time where their peak season is in the early spring. Now for sub-yearling migrants, these fish can look a lot different. And importantly, it was believed to only be found in 2 populations of Fraser River sockeye. Now, these fish differ because they do not have a period of freshwater development. So these fish are born in freshwater and emerge from their eggs, but they immediately begin moving to the estuary, and that's the area that they have this kind of growth and development that they need for the ocean. When we think of development that fish need for the ocean, we can think about their body size first. So if they're going to be eaten by too many things when they get to the ocean habitat, but also the diet. So when fish are in a freshwater environment, they're mostly going to be feeding on bugs or other invertebrates, whereas in the ocean, they need to be able to feed on other fish, and that will expand the range of things that they're able to feed on and then survive. So as I was saying, these fish move to the estuary right away, and that's their site of growth. They're smaller bodied because they don't have that period of freshwater development, and they have a later peak migration. So these fish travel more so in the late spring or early summer. So my research is happening all over the lower Fraser River, and I did my sampling for my master's in the summer and spring of 2023 and 2024. The area that I sampled was a really huge area. It was super expansive, and that's because the tidal environment of the lower Fraser River is really far-reaching because it's so flat. So that means that there's a lot of mixing between the freshwater and saltwater, which is characteristic of the estuary habitat, and that's where my research is based. So at each site that I go to, I had, I think, 58 sites in my master's project that was sampled by myself and also other teams for a conservation group that I am partnered with. And we're looking at the fish communities that are there. So that's fish that are salmon and also non-salmonid species, because we're examining any fish that's in the estuary. So to capture the fish, I use a beach seine, which is a big net that you can walk through the river. Also fyke nets, which sample the tidal cycles and fish that are using the estuary vegetation. So then when it's high tide, the fish are in the vegetation, eating there, resting there, and then at low tide, the fish are cued to move so that they don't get stranded. I also have used a purse seine, and that is in the deeper water sections of the Fraser and primarily in the eelgrass habitat. And that's where a lot of the fish are feeding on invertebrates, but also salmon, because that's the location of the outer estuary, and so they're pretty close to moving out to the ocean for their adulthood. So when I capture a salmon species, I first measure the species or the individual, and I take the fork length, which is the length of the fish from the tip of its nose to the V of the caudal fin or the tail fin. And this is useful to have a standard measurement that can be compared to all fish that I capture. Next, I took genetic clips of the salmon that I found, and these are really interesting because you can use the DNA that's on the paper once laid on. This is a genetic clip of the tip of the tail fin. And so the tail fin tip is taken off, placed on special paper, and the DNA can be read on this paper to find out where that fish is from, so its population of origin, which is really cool because the estuary contains all these fish that are migrating at the same time, but these salmon come from all over the Fraser watershed, which is over 200,000 kilometers squared, so a huge area, and all the fish are moving through it. So what did I get to see in my fieldwork? So I have over 600 fish or sockeye salmon that were identified in the Lower Fraser between 2023 and 2024 in those springs and summers. So this figure here includes both years, where 2023 is shown in red or in blue and 2024 is shown in red. On the horizontal axis is the sampling date that the fish was captured, and then on the y-axis or the vertical line is the fork length measurement. So looking at this graph, we can see that there's a couple of patterns in the data. So the first group that I'll draw our attention to is this group at the top of the graph. And so these, we can see, have a large body size compared to some of the other points, and they were captured early. So this pairing, as well as the fact that they were sampled primarily in the eelgrass, which is that outer estuary zone, indicates to us that these fish are ready for the ocean and then are more likely to be subyearling fish, or at least showing that pattern that we would expect from them. So the next group is the cluster that's down below at the bottom of the graph, where there are smaller bodied fish that are entering the estuary in the springtime, but growing over time and increasing their body size so they can still exit the estuary in the late summer. So this is more similar to what we would expect from a subyearling strategy. But what's interesting is that we previously understood that the subyearling strategy was only in 2 populations of Fraser sockeye, which would make a fraction of the salmon that are moving through that area. So how are there so many data points that are replicating the subyearling strategy? When I add in my genetic information to the capture data that I have, we can start to make some estimates on what's happening here. So this is a similar graph to what was just above or previously shown, but this is for 2023 only, and I've also included my populations as a factor. So on the right side of the graph are the populations that I captured. I had 13 populations in this year. The most popular population was from Shuswap Complex, followed by Kamloops. And here we can see the same pattern that we did before. There's a group of larger bodied fish that are in the estuary early on, and then a group of these smaller bodied fish here as well. So we can zoom into this just to make that a bit more clear and see that there's a diversity of populations that are represented in this subyearling pattern. So now looking at 2024, a similar situation. We have larger bodied fish moving forward in the early estuary or in the early spring, followed by the second pattern of the subyearling strategy. And again, we have a diversity of populations that are represented in 2024 as well. In this year, the most popular population that I caught was from Kamloops, followed by Quesnel. And something of interest is that some populations, including Quesnel, had fish that were represented in both the subyearling and also the yearling strategy pattern that I've just shown on my screen. So this kind of shows that populations may be having some split individuals that are migrating early as a larger body size, or migrating as a larger body size, having done this period of freshwater development, and also some that may be moving through their early development faster to get to the estuary. So this has led me to question if sockeye populations may be expanding their life history strategies to be more inclusive of the subyearling strategy. It also has led me to wonder if we should start managing or discussing sockeye populations more in a nuanced way rather than only at the population level, because there are some differences between the individuals that I've seen in my data. To really understand this, I think that it'd be useful to examine temperature data from the areas that these fish originate from, because as we know that a driver of migration for fish is the environment that the fish is growing up in. If there is a stress in that environment, like a high temperature, then I wonder if the fish are being cued to leave that stress and then go to the estuary and do its development there. However, that's a little bit outside the scope of my project necessarily, but something that I will be looking into as I continue analyzing my data. So working on the Fraser, I've seen a lot of the impact of human activities here, which really showcases the importance of maintaining the environment that is here. So these are just some examples that I encounter in my day-to-day on the field, including log booms, as there's transport on the Fraser, and also construction on the stream side. So all this is impacting fish habitat. But as I've seen, juvenile salmon from both life histories are using off-channel areas for their early development so they can get to the ocean and be competitive. And most importantly, this could be really valuable to sub-yearling fish who are relying on this in a different way than those that are yearling. So I have partnered with a restoration group called Raincoast Conservation Foundation on my research, and they have conducted some restoration work on the Fraser River where my fish are located. So an example of a project that we just worked on is Woods Island, which is a marsh island that is north of the airport. And here we lowered the elevation of marshland that was inaccessible to fish, so that that marshland could then be inundated with water more often with the tide cycles. We created channels so that fish could swim up into the marshland and access back channels as they formed over time. And we also planted species so that fish could have areas of refuge and also would foster invertebrates and things that fish could then benefit from as they're feeding. I'll say it was really cool to get to work on this kind of restoration project because often science is done over really long time frames, which makes a lot of sense, but it was cool to be part of a project where we saw really immediate success. I had been working for Raincoast for a number of years, so I'd done monitoring of the area beforehand to see if fish were in the area and making use of the habitat, and there were very few. And this past summer, I found that there were hundreds of salmon that were using this area, and that's only in the first year of its development. So that's really exciting. So into some takeaways from my presentation. The Fraser Estuary supports two life histories of sockeye salmon, which are the yearling and sub-yearling migrants. It may be possible that more populations of Fraser River sockeye are expanding their life histories to be more inclusive of the sub-yearling strategy, which could be indicated by the fact that there's more populations with smaller bodied fish moving through the estuary and growing over time. Finally, salmon live in urban environments, and restoration and the work that we do is making a difference. And I find it really encouraging that they're making use of the area when we do action to try to assist them in their productivity. So these are my references from my presentation. And thank you so much for coming, and to Coho Festival for having me.